Hi, I am Karen Vanderwolf, a PhD student in the Environmental and Life Sciences program, and today I'm going to be talking about skin chemistry and skin disease in bats. So in humans, skin chemistry is really important in various types of skin infections. Skin disorders such as dermatitis, which is any type of skin irritation, and fungal infections such as athlete's foot can be associated or exacerbated by increases in skin pH. So the reason it happens is cutaneous pH, so skin pH, can alter enzymes produced by pathogens and the host, and also the function of these enzymes. So you may have experienced this with yourself with acne. A common way to treat acne is to apply topical acids to the face. So a really common one is salicylic acid. This makes the skin on the face less hospitable to the bacteria that lead to acne. A study found that trying to experimentally induce fungal lesions on human skin was much more successful for skin that had been experimentally alkalized, so the pH increased, than on normal human skin, which is regularly acidic. So for the pH scale, 7 is neutral, above 7 is alkaline, below 7 is acidic. Human skin is pretty acidic, normally at about 4.5, although this varies quite a bit. Now in bats, skin conditions aren't very commonly reported simply because there is a lack of studies. They do have some forms of dermatitis, which has been recorded in captive bat colonies. However, in 2006, a new skin condition arose, which was called white nose syndrome, which is caused by a fungus. This is a picture of a bat that has died from white nose syndrome. I took this in 2011 in New Brunswick. You can see all the white sort of pinpricks. That's the fungus growing on all the exposed skin parts. So this disease has killed millions of bats in North America. It's caused by an invasive fungus, either from Europe or Asia, that was probably brought over by humans. The first cases of the disease were found in 2006 in a commercial cave in New York State. It only affects bats during the winter when they're hibernating. I'll go into that a little more later. But not all bat species are affected, and even within species that are affected, not all individuals perish from the disease. So I wanted to know why that is, and I thought I would focus on skin chemistry to see if different bat species and different individuals had different skin chemistries that might influence the progression of the disease. So my first step in this study was, of course, to look at the literature. Was the skin pH of bats already known? And actually, I was surprised to find that there is very little information out there about skin pH of animals. There is, of course, the greatest information available for humans, and then domestic animals and lab animals. But I really couldn't find any study on any wildlife species, let alone bats. So this was a big knowledge gap. And among the species for which there is information, you can see there's quite a range there. So humans, quite acidic, and then it goes up from there to sheep, which are quite alkaline. So that was quite interesting. And I wanted to know where bats fit in to this scale. So the objectives were to measure 5 bat species in Canada and determine how the wing skin pH varied among the different bat species, between the two sexes, among seasons, and also different body parts, so different parts of the wing and geographic sites. I also had access to a captive bat colony in Hamilton, Ontario, which I measured once a month for 1 year. So how do you measure skin pH? Well, what I did is I took a pH meter that was designed for humans for dermatology studies, and I used that on the bat skin. I took 6 measurements per bat, so on different areas of the wing, to try to get that variation. And in the end, I measured over 700 bats in Ontario, Quebec, New Brunswick, and PEI. And I found that there was quite a bit of variation. At the very top there, the sort of black and white bat, you can see all the places on the wing that I measured. I didn't measure all of those dots for all bats, simply because logistics. It takes a long time to take that many measurements. And I found that the places where you get the most variation were sort of the empty circles there with the APU. Those were the ones I measured for all of the bats. So I have displayed here 2 species, the Myotis lucificus, which is the little brown bats, and then Pathogens fuscus, which is big brown bats. And you can see there's quite a bit of variation here. So the light colors, the light yellow, is more alkaline, higher pH, and the dark colors are more acidic pH. So you can see the bat at the very bottom is quite dark, quite brown even. That is a captive big brown bat, and I really found quite a difference between captive bats and wild bats, which I'll go into later. But you can also see there's variation across the wing within individuals. So there's a lot of information on this graph. On the y-axis, there's the skin pH values, and then along the bottom axis, the x-axis, we have the 5 different bat species that I measured. So to focus first on hibernation. So essentially what I'm presenting here is the 3 biological seasons for bats. Hibernation during the winter, of course, they essentially go to sleep in caves and they don't eat for the entire time they're in there because they only eat insects and insects aren't available. So they decrease their metabolism to survive this period of famine. And you can see that among the bat species that I measured during hibernation, there's quite a difference in skin pH. The captive bats are much more acidic. They're the ones with the most measurements because I had constant access to them. And the wild bats are a lot more alkaline during the hibernation period, so during winter. And this varies among species as well. So the big brown bats were more acidic than the other species that I measured. During maternity season, so this is from May to July, when the pups are born and raised by the females, skin pH goes down for all species compared to the winter. So they're more acidic in the summer than during the winter. Again, there is variation among the bat species that I measured, and the big brown bats, particularly the captive big brown bats of Pathogens fuscus, were more acidic. During swarming season, this is mating season for the bats, which is during August to October, so just before hibernation, skin pH begins to rise again. But I am seeing the same sort of species pattern that I saw during the other 2 seasons. So the captive big brown bats, again, a lot more acidic than the wild bats. But the wild big brown bats are also more acidic than the other bat species that I measured. And this graph just gives you a better sense of seasonal variation. So again, skin pH is shown on the y-axis. On the bottom axis, the x-axis, I have day of year. So January 1st is day 0 of the year. Day 200 is in July, just to give you a sense of time on this axis. So you can see that it's quite a curve here, sort of a bowl shape curve of bat pH over time. And this is quite similar to humans, actually. We also get more acidic during summer, during July particularly, and more alkaline, so higher pH during the winter. You can see this pattern is also seen in the captive bats. So in the top graphs, it's that really long bowl shape. But the seasonal variation seen in the captive bats is attenuated, so there's not as much variation as with the wild bats, which is not too surprising given that the captive bats are in a somewhat climate-controlled environment. And the reason I think the captive bats are so much more acidic than the wild bats is because of where they're roosting. So this is the captive big brown bat colony. You can see they like to roost in these towels. And I'm sure you can also notice that the towels are kind of gross. They're covered in guano, so bat poop, and they are also, perhaps you can see, a bit stiff because the bats are peeing on the towel and the towel gets stiff with the urine. So bat urine is quite acidic. It's about 5.5 pH, so that's below the 7 neutral. And I did measure the pH of the towels, and a surprise, it was also quite acidic. So the bats are roosting on this really acidic substrate, which seems to be acidifying their skin, which does not occur, of course, in the wild bats because they don't roost in towels. Let's see if I can get out of this fade. So as I mentioned, I measured the pH of the towels, and the towels, when they were dirty, were 5.6. Clean towels are neutral at 7 pH. In the captive colony, the bats also have access to a wooden structure to roost in, and in one part of the colony that is heavily used, the wood was 6.6 pH, so acidic. But the wood in a different part of the colony that was not used very often was 8 pH, so it was actually alkaline. So when the bats are using the structure a lot and peeing on it, it's making it a lot more acidic, which then has a feedback with the skin pH. I did not measure cave walls during hibernation, but in the literature, other people have measured the pH of cave walls, and it's quite alkaline generally, depending on the cave, about 7 to 8 pH, which might partially contribute to the fact that the bat skin is so alkaline during hibernation, during winter when they are in these caves. So the graph is showing individuals that I measured over time in the captive bat colony. So again, skin pH is on the y-axis and day of year is on the x-axis on the bottom. Each color is an individual bat that I measured repeatedly over 1 year. Now, on the second to last points, that's in November, you might notice there's quite a big spike there. So skin pH is increasing in these individuals. Just a week before I went to the colony, the towels that they were roosting in were changed for clean towels. And so you can kind of see the response of the skin pH to those clean, neutral, or even alkaline pH substrate that they were roosting in. So it really seems to have an effect on their skin pH. However, it's not just roosting substrate that affects your skin. There are also other factors, environmental factors, temperature, humidity, and other skin factors such as the amount of sebum or the amount of lipids you're producing on your skin. So certainly more things to look at in the future. So I found that skin pH varies among individuals, within individuals, and among bat species. But so what? Why does this matter? The reason is not just because skin pH affects enzyme function, but also seems to affect what sort of microbes are found on skin. So on bats, there's a variety of fungi and bacteria on their skin, and it's a substrate of microbes that are found in their environment. So the environment on the skin is going to partially determine what microbes from the general environment can survive on the skin and grow on the skin. This includes potential pathogens. So in humans, if you temporarily acidify your skin with topical products, that will decrease the bacterial diversity found on your skin. Conversely, if you increase your skin pH, you can increase your bacterial diversity. And presumably something like this might happen in bats. So for the future research, I definitely want to find out more about how other aspects of skin chemistry relate to pH and influence pH. Certainly the amount of lipids on the skin and how all these aspects affect disease progression. So at more acidic pH, for example, does the pathogen that causes white nose syndrome not function as well? So the enzymes that it produces, do they function at all? So I kept mentioning that the big brown bats have the most acidic skin. And of the bat species that I measured, they also survive white nose syndrome, the skin disease, the best. They have the lowest mortality rate compared to the other bat species like little brown bats. Perhaps skin pH does have something to do with this. Or perhaps it's an indirect effect, not a direct effect on the pathogen. For example, lower skin pH can also help with cutaneous water loss. So if you have acidic pH, you generally don't lose water from your skin as fast as with more alkaline pH. And this can help a lot with surviving diseases like white nose syndrome, where one of the mechanisms by which you die is from dehydration. So too much water loss from such compromised skin. So that concludes my presentation. I would like to thank a lot of the people which are listed here, especially my committee, Christina Davey and Christopher Kyle, and of course the funding sources such as M&R, Trent, and the New Brunswick Museum. Thank you for listening.